Thank you for taking time to see my research presentation. Melanin synthesis is a complex process. It starts with the development and differentiation of melanocyte stem cells, which have to be properly migrated and delivered to the skin. And within these cells, a multitude of proteins are involved in maintaining an ideal environment and transport of proteins and enzymes for melanin synthesis. Once melanin is synthesized, these have to be properly transported and delivered to the skin, hair, and plumage. I chose to work with mc1r, the melanocortin-1 receptor, because it has been shown in several vertebrates to be involved in red pigmentation. It sits on the membrane of the melanocyte and directs the pigment synthesis toward black pigment, eumelanin, or red pigment, pheomelanin. When it is hyperactivated, it causes black pigmentation. When it is inactivated, it causes red pigmentation. It also interacts with alpha MSH and agouti on the exterior of the cell. When alpha MSH is hyperactivated and agouti is inactivated, you will again get black pigment. If alpha MSH is inactivated and agouti is hyperactivated, you will get red pigment. In pigeons, there are two traits that fall at the extremes of this red-black spectrum, the spread black trait and the recessive red trait. There are other darkening traits as well, but I chose to start with this. mc1r in birds is a single exon gene. I designed 3 amplicons spanning the gene in order to PCR amplify. However, the middle amplicon failed only in red birds. The sample included red, blue, and black birds. So I redesigned the PCR amplicon, shown by the arrows, and found that there is a polymorphism here in red birds and in black birds. So to test this further, I went to the bioinformatic data of the available genomes and found that several genomes have this deletion starting at the same coordinates shown by the black strips in 6 of the columns. I tested every red bird in the colony or their parents, and only one red bird was lacking any copies of this potential deletion. Sequencing showed that the deletion is exactly 500 base pairs, which is not divisible by 3, and therefore this is a frame shift mutation if it is real. To test this further, I examined a 2-generation pedigree. Normal Mendelian segregation shows that this is not a pseudogenized copy of the gene. mc1r is clearly dead. This is an intragenic frameshift deletion, and it is not recessive red. One thing literature and breeders told us is that smoky, an autosomal recessive trait, is enriched in show red birds. Smoky is a complex syndrome caused by a single gene. However, diagnosis requires several characters that have variable expression, including lightened beak, smudgy wing bars, and lack of a white strip on the tail, as seen in the image on the right. With experience, this can be properly diagnosed, especially in pedigrees. I had 2 pedigrees that were segregating for smoky and were also segregating for the mc1r deletion. Testing every individual in the pedigrees showed complete segregation and a very high combined significance of 5.68 into 10 power minus 13. This shows that this deletion does cause smoky. There is also a cost to being smoky. Until now, from mammalian studies, we thought mc1r can be mutated easily because it is a terminally expressed gene, that is, it's expressed only in melanocytes and nowhere else. However, enrichment of smoky genotype in the unhatched eggs says otherwise. Interestingly, we had told our erstwhile collaborators about this mc1r deletion. In spite of this, they went ahead to publish that another variant, methionine 85, which causes black pigment in two other bird species, they said it causes red in pigeons. They were most likely unable to detect this deletion allele, and the genotypes must have been skewed. Methionine 85, coincidentally, is also segregating in this pedigree. And every individual that is not smoky is dirty, which is an autosomal dominant darkener. Now the probability of every non-smoky bird also being not dirty is extremely low to occur by chance. This can occur only under two scenarios. If, on the one hand, mc1r causes dirty and causes smoky, or if dirty is fixed in this pedigree and only smoky is segregating. In other words, two genes. And these two genes that we would test would be alpha MSH and agouti. However, the experiment is far simpler. What I did was I took a smoky bird from this pedigree and crossed it with an unrelated bird that was neither smoky nor dirty. Taking a one-gene model, we have to use different symbols, and under our model of genotypes, only the smoky allele and wild type allele would segregate, therefore every offspring should be wild type. This would prove allelism. Under a two-gene model, a two-locus model, every offspring should be dirty, and this would suggest that alpha MSH or agouti underlies dirty. What we showed was every offspring that was produced from that cross, images not shown here, were neither dirty nor smoky, and therefore mc1r causes both traits, and therefore methionine 85 causes the dirty phenotype. Not only that, we have also shown that loss of function of mc1r in pigeons does not cause red phenotype. Another result we discovered was the second pedigree had a different allele, non-synonymous mutation in mc1r that was also associated with dirty, because in that family too, every individual that is not smoky is dirty. In conclusion, there is a severe deficiency of null alleles in birds, suggesting that there are fitness consequences from mutations in this gene, not just for pigeons, but in birds. And there's a deficiency of hypomorphs, in other words, red pigment in birds. This is because researchers have been missing association because they've been trained on the mammalian understanding of pigment synthesis. Finally, the lack of red phenotype in mc1r hypomorphs is quite likely because other genes are participating in this process and because the feather morphology creates a more complex environment. With that, I would like to thank the funding from the University of Texas at Arlington to Dr. Fardin, mentoring by Dr. Fardin, interns and breeders for the help in discussions. With that, I'd like to thank you all.